Hey guys, welcome back to Unstop Biology. I hope you guys are doing good and your preparation is going on very well. So we don't have much of time left, guys. Let's finish off the NCERT for class 11th and 12th as soon as possible. So guys, before we start off with this video, please let me know with your comments, with your feedbacks, how are you finding my videos? If you need any kind of improvement, I'm telling you because I want to make the best of videos for so that you can understand these topics in a very easy way. Right now we are covering with the NCERT and we are doing NCERT class 9th for no, sorry NCERT class NCERT for class 12th, and we are at chapter 9, right? So in the last video, guys, we finished off the topic of animal breeding, right? So basically this chapter is all about the strategies for enhancement in food production. And what we have studied in animal breeding is that how do we actually improvise the quality and the yield, increase the yield by using different kind of animal breeding methods. So in this video we are going to cover plant breeding. We are going to see what exactly is plant breeding, right? So guys, if you remember about the green revolution, if you have ever heard about the green revolution, that was dependent to a large extent on the plant breeding techniques. And here again, what is the aim? The aim is high yielding and disease resistant varieties in various crops, right? So our aim remains the same as of animal breeding. We just need high yield, high quality of crops. So what is plant breeding? It is basically, purposeful manipulation. So basically we are manipulating the naturally occurring plant species in order to create desired plant types which are basically better suited for cultivation, and it gives better yields and are disease resistant. So basically we are purposefully manipulating plant species to get better yield and better quality of crops, right? So basically classical plant breeding involves crossing, hybridization of pure lines of plants, then it is followed by artificially selecting the produce plants, and then these produce basically are with desirable traits of higher yields, nutrition, quality, resistance to disease, and a lot of things, right? So what are the main steps in breeding? Let's see that, right? So in order to basically breed a new genetic variety of a crop, the main steps are first collection of variability, right? So what is collection of variability? Basically genetic variability, variability in diversity in genes, right? So genetic variability is basically root of any breeding program. First, you'll have to select the best genetic variable of a plant, right? So in many crops, the pre-existing genetic variability is available from wild relatives of the crop, right? So it is basically available in the wild relatives of the crops. Now collection and preservation of all these different wild varieties, right? Their species, their relatives of the cultivated species is a prerequisite for effective exploitation of natural genes, right? So entire collection of plants or seeds, what we do is we collect these wild plants or their seeds, and this entire collection of plants or seeds having all diverse alleles. So what I mean, entire collection of what? Collection of all the diverse alleles for all genes in a given crop is known as germplasm, germplasm collection. It is known as germplasm collection. Okay, so first we collect all the genetic varieties of the plant or seeds. Then what we do? Then we evaluate and select the parent crop, right? The parent plant. So basically, then this germplasm collection is evaluated to identify plants with desirable combination of characters. So what kind of yield we need, as per that we choose the parent, and these selected plants basically are multiplied and used in the process of hybridization. So basically, where we need pure lines, we create pure lines. Where we need hybrids, we create hybrids. Then what we do? So after creating these hybrids, we basically start selecting and testing it for superior recombinations. So now basically we have a progeny of hybrids where there should be desired characters of combination, right? So now what we do is we select these from these progeny, we select the ones which are actually the best suited as per our requirement, and then what we do is we start self-pollinating them. And we self-pollinate them for several of generations till they reach a state of uniformity of homozygosity. So guys, it is basically very similar to what we read in case of animal breeding. What we discussed in case of animal breeding, we discussed that after getting the superior progeny, we used to basically grow it or reproduce it for 4 to 6 generations. And why we used to do that? To achieve homozygosity, so that it has all the characters in it which are required. In the same way, in plant breeding, we in order to reach homozygosity, in order to for it to have all the characters which are required so that it does not segregate, we keep on self-pollinating it for several generations. And what we do after self-pollinating it for several generations, we again test it and then we release it. So basically, how is the testing done? It's not again so simple, right? We basically evaluate the new lines, right? We evaluate it as per the yield, the quality, disease resistance, and all that. Once we see that yes, it's good in all those things, then what we do? We grow it in our research fields, right? We keep growing it in our research field. We record their performance under different type of fertilizers to understand what is the ideal fertilizer to be used, what kind of irrigation system to be used, what are the crop management practices to be used. And after that, we test it in the farmer's field, right? So in different locations in the country, we test it because we have different seasons in different parts of India, right? So at different locations, we test it in the farmer's field, right? And after we test in the farmer's field, we start growing it in the commercialized way. So basically, we then release it and commercialize it, right? So basically, what happens is, in India, there are several varieties of wheat and rice. These varieties of wheat and rice basically were developed in mid 1960s. This time is known as the time of Green Revolution. That is why it is known as Green Revolution. So basically, we got a lot of plant breeding techniques, and we kind of revolutionized the the older way of growing crops. So what we did, like in case of wheat and rice, in the period between 1960 to 2000, the wheat production increased from 11 million tons to 75 million tons. In the same way, rice production went up from 35 million tons to 89.5 million tons. So basically, as our population was increasing, the demand for these basic crops were increasing, and In order to do that, it was not possible through the authentic, the older way of agriculture. We got a lot of high yielding plants, right? So basically, the semi-dwarf varieties of wheat and rice were developed. So we took varieties from Philippines, from Taiwan, and then we grew our own dwarf varieties, right? In the same way, sugarcane. So basically, what happens is Saccharum barbary was originally grown in North India, but it had very poor content and yield. Now, if you see the tropical canes which are grown in South India, which is Saccharum officinarum, right? That had thicker stems but light sugar content, sorry, thicker stems and higher sugar content. But then it was not possible to grow them in North India because of climate conditions. Now, what happened is scientists they crossed these two species of one of North India and one of South India, and they combined the desirable qualities of high yield, thick stem, high sugar, and ability to grow across North India. And then they supplied it, they commercialized it. In the same way, millets के साथ it happened that hybrid maize, jowar, bajra, everything was successfully developed in India, right? So what is the significance of plant weed breeding for disease resistance? We'll see that as well. So basically, guys, a wide range of fungal, bacterial, viral pathogens affect the yield of cultivated crop species. So basically, if we talk about crop losses because of this, it is almost 20 to 30% at least, to even 100%, right? So you can imagine, how difficult it is with all these, pathogens, even plant pathogens, to cultivate crops. So basically, there were breeding and development of, hybrids which are resistant to disease, so to these diseases, and that enhances the food production, the crop production, right? So resistant of host plant is basically ability to prevent the pathogen from causing disease, and again it is determined by genetic constitution. So you can see that the genes play a very important role everywhere, right? Now, what are the methods of breeding for disease resistant? So basically it is carried in by the breeding technique, by the conventional breeding technique of hybridization and selection that we studied already, right? There are some by mutation breeding as well. Now, the various sequential steps again will be like you will screen the germ plasm, you will hybridize the selected parent, you will select and evaluate, you will test, and you will release the new variety. If you see the table, here you will see that wheat, brassica, cauliflower, cowpea, chili, like all of these, varieties of plants are resistant to diseases. So you see the variety and then resistant to disease. So leaf and stripe rust, white rust, black rot, curl blight, right? So many diseases are there. Now basically inducing mutations in plants. So other form is mutation, through mutation, right? So inducing mutations in plant through diverse means and then screening the plant material for resistance. This also leads to desirable genes being identified, right? So basically, plants having desirable characters can then be either multiplied directly or they can be used in breeding, right? So basically, we have heard about mutation. Mutation is nothing but genetic variation which are created through changes in the base sequence of gene, right? So it is possible to induce mutation artificially through say use of chemicals, radiations, by selecting the plant that have desirable characters as source of breeding, right? This type of breeding is known as mutation breeding. So in moong bean, resistance to yellow mosaic virus and powdery mildew were induced by mutation. So remember the example for your boards exam, right? Now, what is basically plant breeding for developing resistance to insect pests? So basically, in the case of plant, guys, what happens is that it's not just pathogens, it's not just bacteria or viruses. There are so many insects which depend on these plants. So basically these insects become pests on these plants, right? And they destroy the crop completely. So insect resistance in host crop plant is also required. Now this resistance may be due to morphological, biological or biochemical or physiological characteristics, right? So say example is resistance to jacids in cotton, right? Or cereal leaf beetle in wheat. Now what happens is that source of resistance genes. It may be cultivated varieties, germplasm correction of the crop, or wild relatives, right? So basically, the breeding methods for insect pest resistance will be the same as we discussed before, right? So basically, it will be the conventional breeding method or the mutation breeding. So if you'll see here again another table. So in the case of rapeseed mustard, brassica, you have a variety which is Pusa Gaurav, and the insect pest is aphid, right? So aphids are there which attack brassica crop, right? So again, it is the same, same type of breeding method if you want to develop resistance against pests. Then plant breeding for improved food quality. Now if you want to improve the quality of food, right? So basically with say higher level of vitamins, minerals, or whatever type of biomolecules that your body requires, right? So you can even breed plant for improved food quality, right? Now what we do is we biofortificate, right? So biofortification is the most practical means to improve public health. Basically, you know, increasing the levels of vitamins, minerals, proteins, or healthier fats by plant breeding. And breeding for improved nutritional quality is again undertaken with the objective of improving protein content and quality, oil content and quality, vitamin content, and micronutrient and mineral content. Guys, we have certain examples for these. Say there is a maize hybrid. So in 2000, maize hybrids were created, right? That had twice the number of amino acids, which were lysine and tryptophan. Right? So maize hybrids were created which has double the amount of amino acids, lysine and tryptophan. Right? In the same way, there are many crops which are developed from the perspective of improving food quality. Like there are rice varieties which are rich in vitamins and minerals, right? So guys, with this we have finished plant breeding as well. I hope you understood this topic very well. So please do like the video and share it with your friends as much as you can. And if you're new to the channel, as I say always, please subscribe to the channel and press the bell icon to get all the interesting updates. And guys, if you have any doubt in regards to this topic, please drop in your doubts in comments. Please tell me how do you like the video, or do you have any problem? Do you want to change something in my videos? Or you can also message me on WhatsApp. The number is given in the description of the video. Okay guys, thank you for watching this video. See you in the next video. Bye bye.